Yeah, perfect. Brilliant. That's all I needed to know. So, session 3, we're still looking at cells, tissues, and organs for healthcare, and we'll have one more session on this unit next session. And let's just pop that up. That's the unit that we're on. And what we need to do today in this session is have a look at some of these tissues and how the cells that we looked at last session make up some of these tissues. So I'm going to recap briefly to put you back where we were last session. Let me just, that's what I need to do. Get into slideshow mode. Oh, wrong one. Slideshow. Let's go. That will make it a little bit clearer for you. So I realize there's not many people because it's Saturday, probably people have got things to do, and most of you are probably watching the recording. So what I would like you to do is have a look at these questions and have a think about the answers. Oh, I've got them labeled 1, 2, 3, 3 there. That should be 1, 2, 3, 4, obviously. But what I'd like you to have a look at is think about the answers, write them down, and then you'll be thinking about the stuff that we did last session, hopefully, and be able to move along. So I'm just going to pause for a minute there. If you want to put them in the chat in the lesson, that's absolutely fine. I can have a look at your understanding, but if you don't want to and you feel like you'd rather just do it on paper, that's fine too. So I'm just going to give you a minute to have a look through these, to have a think which you think's the correct answer, and then I'll pop them up on the screen for you. Number 1 for the last one. The second, number 3. Okay. I've copied and pasted that across from another slide and not changed the number there, I think. So apologies for that. We can call it number 4. We know what comes after 3. So number 4, you're going for number 1. We'll look at the answers you're giving me. Okay, we've got some good answers coming in, some correct, some not quite correct, but it's making you think. That's the main thing. So good work, you lot. Well done. Oh, good, good, good. Some really good answers there, guys. Well done. I'm just going to pop the answers up on the screen for you, and you can self-check, obviously. So, which type of tissue would you expect to find lining the trachea? That's the ciliated epithelium. Remember, we've got that sort of wave-like motion of these sticky out bits of the cell that helps to move the mucus over the trachea we were talking about. Which of the following statements is true? Voluntary muscle looks stripy under the light microscope. So voluntary muscle is also known as striated muscle or skeletal muscle. That's the one that you can move voluntarily, like your legs, your arms, your eyes, things like that. The involuntary muscle, which is also known as smooth muscle, smooth because it's not stripy, is what lines the gut. And which of the following definitions is a definition of tissues? It's a group of cells, not necessarily identical, but from the same origin, that together carry out a specific function. So we had a little look at the cells and some specialist cells of certain tissue types last session. And we're going to have a look a bit more how it's held together, well, not so much held together, how it works together as tissues now, these main cell groups. One cell group we didn't get round to having a look at last session was neurons. We had a look at epithelium, we had a look at muscles, we had a look at connective tissue, and we need to have a look at neurons as well. So nervous tissue is, it's not just neurons, it's nervous tissue we're going to consider, but we really focus on the neuron. So nervous tissue is made up of two types of, oh, sorry, I've got my phone talking to me there. Don't know why, it just kicked in the AI. So nervous tissue contains two types of cells, neurons and glial cells. And there's a nerve cell there. You can see it's nervous tissue. That's what they look like. We'll have a look at the structures in a moment, but this is the nerve cell here. And you can see it's got all these projections, all these bits that stick out away from the main body of the cell. The tissue itself is really important because it can send and generate electrical impulses. They're transmitted down the spinal cord and out to the body. So down the spinal cord, then out to your arms, to your legs, and out to the periphery. And we'll have a look at how that's structured as a tissue later on today. So here we have our first glimpse at a neuron. This is a nerve cell, and this is what forms the tissue, these neurons that transmit the information through the central and peripheral nervous system. So central means the middle, central, peripheral on the periphery, the outside. So the central nervous system is literally what is in the center, your brain and your spinal cord. The peripheral nervous system is what goes out down your arms and your legs and around your body. So we're going to have a look at this, these cells, and then have a look at it as a tissue. Now, some of this information that I'm going to give you is going to sound a bit confusing and hard, and this next statement is one. We are going to unlock it and understand it later in the course. So don't worry too much about this now. It is hard and tricky. But what happens is the information gets sent along the neuron. So this is a neuron, the nerve cell, and the information, it's an electrical impulse that gets sent along, and we call that an action potential, that impulse. Don't worry too much about that term for now. We will explain it later in a later unit of this course. And it causes something called a wave of depolarization, which moves along the nerve cell. That sounds quite confusing. It changes charge. What happens is this is charged. So think like static electricity is a charge. And the charge moves along this like an electricity moving along an electrical cable. So the charge moves along that neuron. That's what happens. Sort of this as a summary of what's going on there. So we're going to have a look at these neurons themselves. So this is a nice clear drawing of the neuron, and here we've got sort of simplified version of it at the top. So there's different structures, as you can see. It's the main bit of the cell there. If you look really closely, you can see some of the structures we've talked about over the last couple of sessions. There's mitochondria in there. There's endoplasmic reticulum in there. There's a Golgi apparatus in there. There's a nucleus in there. It's just like a normal cell, like we would expect. All these structures we've talked about. But then we've got all these branches coming off. And these are called dendrites. So anything that's called dendritic means tree-like branching. It's a very common structure to see in nature, you know, in trees, for example. The reason for that is it allows branching and covering large surface areas. So trees branch out to maximize the amount of light they get, so the leaves can photosynthesize. Neurons branch out to maximize the amount of connections they can make. So it's a very familiar structure in biology. So we've got this branching, this dendritic structure, and these are called dendrites, these branches essentially that come off the neuron. And the important bit of the dendrites is their tips, because each one of these tips can form a connection with another neuron, the branches of another neuron. And that's how we send communication signals around our nervous system. So each one of those dendrites at the end has something called a synaptic terminal. And that's a connection with a new neuron. I mean, here it says a presynaptic terminal. So terminal, the synapse we're going to learn about much later on again is the gap between two neurons. So you might see it called a presynaptic terminal, which means before the synapse, or you might see it labeled as a postsynaptic terminal, which means after the synapse. But you're more likely to just see synaptic terminal. Just means the end is where the connections are made between two neurons. So the dendrites are how the neuron passes and receives the information between itself and the next neuron. So what else have we got? We've got the cell body, or the soma. It's basically where the signals are interpreted, and the neuron decides, not literally decides, but it's not like a decision, they're not thinking like that, but the neuron will either fire or not fire, depending on if the signal is right, if it's big enough, if it's built up enough. We call this summation, if it's a big enough signal to make it fire. It contains all the organelles, as I just said, things that we recognize from the earlier sessions that we did, the first session, those structures, those cell structures, surrounded by the cytoplasm, which has unfortunately got its own name again, but you know, use the term cytoplasm, everyone knows what you're talking about then. So the axon then is often bundled together with other axons. And when you've got, that's the long, thin piece that could go all the way down your leg, that axon, if it was a nerve in your peripheral nervous system on the periphery on the outside. So the axon could be really, really long. And they're often bundled together with lots of other axons from other neurons. And when they're bundled together, they make up a nerve. And it extends out from the cell body or the soma, and it carries the impulses wherever they've got to go, all the way to your fingertips maybe, or all the way to your toes. So these axons are surrounded by something called myelin. You can see it up here in green. That's the myelin. It's called a myelin sheath. It's a lipid, it's a complex lipid, and it can either it basically insulates the neuron, and it can allow the signal to travel between these myelin bits. There's something called the node of Ranvier there. There's these little gaps, and it allows the neuron to jump, and it allows it to travel really quickly. So rather than the signal going all the way down the nerve, it allows it to jump between these gaps of myelin and speed up the transmission of that nervous impulse. So call it an impulse rather than a signal or a wave or anything. That's the correct term. Call it a nervous impulse. Okay, what else have we got? Synaptic terminal, which I've already mentioned a little bit, is the end of these dendrites, and they've got loads of vesicles, or vesicles if you prefer, and these contain neurotransmitters. And it's the neurotransmitters that form the connection between two neurons. That's why they're really important. You'll have heard of people talking about neurotransmitters in your brain in terms of, I don't know, mental health and things like that. People often talk about them in terms of the effects of drugs on the body, for example. So those terminals release neurotransmitters to the nearby neurons. So they're pretty important structures. Okay, let's have a look. A couple of questions. What is the primary function of the nervous tissue? Which part of the neuron receives incoming information? And what is the function of the axon in the neuron? Have a little go at those and see how much you can remember. I'll just pop the chat up. If you don't fancy putting it on the screen, that's fine. You can just leave it on your rough paper, but do read through and have a go because it will help you with your understanding. Oh, number 2 is a bit tricky, actually. I'd say there's 2 answers, which are kind of okay on that one. Okay, good work. I'll give you a sec. Won't move on too fast. So I'll pop the answers up then. So the function of the nervous tissue is to sense stimuli, stimuluses, if you prefer, and transmit signals through the body. So we sense it with our eyes, our ears, our taste buds, our smell, our fingers, our skin, where we can sense pressure and temperature. Which bit of the neuron receives the incoming information? It's the dendrites at the synaptic terminal sometimes. So B or C is kind of okay there, depending if it's a sensory neuron, which we'll talk about a bit later on. What's the function of the axon? It is to transmit the impulses away or along the nerve, away from the body of the nerve to another cell. It's usually another neuron, or it could be a cell that does something like a muscle. That's an effector cell, which we'll learn about in a bit. Oh, a couple more questions. What's the significance of the nodes of Ranvier along the axon? And what happens at the synaptic terminal of the neuron? So these are quite specific questions and a little bit hard. So see if you remember what these 2 structures are, and then we can move along. I'll give you a minute. Good work, guys. Well done. Pop the answers up. Well done. So the nodes of Ranvier are gaps in the myelin sheath. So the myelin sheath is insulation, a bit like plastic around a electrical wire, but it's got gaps in it. And these gaps are known as the nodes of Ranvier, and they allow impulses to jump, and it really increases the speed of transmission. And the neurotransmitters are there to excite or inhibit the next neuron. That's what happens at the terminals. They pass the message on, the impulse on. Okay, so that was the last type of cell that we needed to look at. And we're going to have a look at tissue now, made up from these cells, these types of specialist cells that we've had a look at over the last couple of sessions. So I'm going to start off by going over this we did last session, and this is just to remind you. So we had a look at a specialist muscle cell. It was a striated muscle cell, which means stripy. So these are the skeletal, striated, or voluntary muscles, 3 names that are used interchangeably, unfortunately. So stripy or striated, voluntary or skeletal. So these are the muscles, like the big muscles on our skeleton that move our limbs, you know, the things we can move voluntarily. And we had a look at the cell structure and basically said they're multinucleated. The striation or stripes is basically due to the presence of these actin and myosin proteins. They're contractile, and that means they can pull in like that. So you've got a muscle like this, and it shortens and thickens when it contracts. thinking about your bicep, it goes from being long and thin to short and thick when you contract it. So that's how those contractile proteins work. And we talked about this last session. They're multinucleated. That's just because of how they were formed, made out of several cells. And now we're going to have a look at how this type of cell makes up tissue. So muscle tissue can be divided into three types. There's the skeletal or stripy, striated, voluntary muscle that we had a look at a specialist cell for last session. We have got cardiac, which forms the heart muscle. That's quite specific. And we've got the smooth muscle. It's just called smooth because it's not stripy. Like that's the opposite of stripy under the microscope, I guess, when they first had started looking at these tissues. So smooth muscle you'll find in things like your guts. There's obviously there is muscle in your guts, but it's not, you haven't got control over it. You can't think, oh, better churn my stomach now to digest my food. It just happens. It's involuntary. So muscle tissue contains these proteins that we were talking about last session, these actin and myosin fibers that slide past one another to allow the movement. And the voluntary muscle tissue, the voluntary contraction allows big movements, the large gross movements that can move us from place to place, like movements of the quadriceps and hamstrings, those sort of muscles, but also finely controlled movements such as the eye as well. And these muscles will look stripy under the microscope, as we've said, because of this actin and myosin fiber structure. That's a micrograph image there of the muscle tissue, and that's what they say looks stripy. Now you might look at that and think, where on earth are the stripes? But if you look over here in this region, you can just about make it out. But to a biologist who's used to looking down the microscope, they'd be like, oh, that's visibly stripy tissue. It would be very clearly stripy to them. I know it doesn't look particularly stripy to like an untrained eye, but these guys would have spent a lot of time looking at tissue. So we also have this smooth muscle tissue over here. These are controlled automatically. So like the gut, we're going to talk in, I think it might be the next unit we're doing actually on digestion. We're going to talk about how food moves automatically down our gut. It's actually the contraction of, because think about your gut, it's mostly tube. So long tube, it called your esophagus, goes from your mouth to your stomach. And food moves down there, it doesn't just drop down. There's circular muscles which squeeze, and there's longitudinal muscles which go lengthwise. So the longitudinal muscles will relax and the circular muscles above it squeeze, and the food gets pushed down, a bit like if you're squeezing a tube of toothpaste. That's how that works. But it's all automatic. You don't think about it. It happens voluntarily. So we'll find these in this smooth muscle, in the digestive system, as I've said, but also we have it in our reproductive organs. We have it in our blood vessels, which is quite important because that means there's some control over the size of the lumen of our blood vessels. Remember, I said lumen means the hole in the tube. So there's a lumen in our gut, actually, since we're speaking about that, and our blood vessels. So we can control the diameter of the hole that goes through the middle of our blood vessels. So that's under muscular control. So that's really good. It means we can increase blood flow to certain areas, and that helps us to cool down our blood if we get too hot, for example. That's why we get red when we get hot, because we increase the blood flow to our skin using these involuntary muscles, for example. And then we've got the cardiac muscle. That's only found in the heart. It's very specialized. It is involuntary, but it's classed differently to the smooth muscle because it's got structural differences. And one of the main differences is we call it myogenic. It can beat, it can generate its own heartbeat. It's not under nervous control, or otherwise, if you got paralyzed, you'd die, your heart would stop. But it can generate its own heartbeat, and we'll look at that in a later unit as well. Okay, so let's have a look at this. What are the function of muscles in the human body, or which is a function. Well, actually, there's 2 on number 1 that are correct. Which type of muscle is involuntary and found in the walls of skeletal organs, and what happens to a smooth muscle when it contracts? And I'll just get my chat up so I can see what you guys are saying. Okay, good work. So I'll just pop those answers up then. So the first one, B, is, oh, it does say pumping blood only. So no, it's not pumping blood only, but it is pumping blood. And C is correct, enabling the movement of the body. That's a function of the skeletal muscles. What type of muscle is involuntary? It's smooth. It just doesn't look stripy, basically. And what happens when the muscle when it contracts? It becomes shorter and thicker. Those fibers pull in together. Oh. Okay, so this is a bit of revision from last session where we looked at epithelial cells, and we said we could talk about it in terms of how many layers it has. So it could be simple with a single layer, stratified with several layers, or pseudo-stratified, as in it looked stratified, but it wasn't. So if you looked at this, it might look like there's 2 layers, but there's not really, it's just one. So that's one way that we would have described epithelial cells. We've got these different cell shapes. We've got the squamous or squamous, I've heard it called as well, where it's flattened and looks like sort of tiles. We have the cuboidal, which is a cube or cuboid shape. And what else have we got? Stratified means it's layered. Yeah, that's what we really need to know there. And we also talked about the surface specialisation, such as cilia, didn't we? And we had a look actually at the goblet cell as a specific example. So we said that the surface epithelia form continuous sheets, often with multiple cell layers. And one of the things that they do is they form tight cell junctions. So they're like tiles on a wall. And this gives a lot of strength and protection against one, mechanical damage. So if you think about your digestive system, that gets quite a lot of a battering because it's an exchange surface. So all of your food is getting churned up against it. You're releasing really strong acids into your stomach section. Proteases, which are very nasty enzymes. They break down protein, which our body is made out of. So it's a very much needed protective layer. It's got a basement membrane, which have a look at this if you think about like a good lining to some sort of structure. Like think how well protected that stratified layer would be. You've got this dense network of cells, they're tightly back together. This really, really important structure. So it forms there's a basement membrane at the bottom, which forms a separation between the underlying connective tissue, which might be, I don't know, it could be anything, could be cartilage, could be fat, could be bone, whatever it's stuck to. And we talked about the fact that they are polarized. There's different ends to the cell. So you've got an apical end, which is like the surface end. So example here on the surface, there's cilia. So they're specialized at the top. And then you've got basal end, a bottom end that forms the membrane at the bottom. We had a look at these specialized goblet cells, didn't we? A special type of epithelial cell that's polarized. Let me just see what's... Would the basement membrane be part of the tissue then? Yes, it is part of the tissue. Yeah, yeah. This is an example of a specialized epithelial cell because obviously they're all a bit different. We talked about the fact that it's polar, so all of the vesicles and excretory vesicles are at the top. The nucleus and the other structures are towards the bottom in this particular example. So it's a very specific example. There's specialist sorts of cells. This would be found somewhere like in the gut, for example, where it's primed for releasing mucins. So it's got all these adaptations for this role, loads and loads of secretory vesicles, increased surface area to release more of these vesicles. So it's good at its job. So what does this look like as a tissue? Well, it covers large surfaces of the body, which are exposed to the external environment. So as we've talked about, the line in the gut is really important, all the inside of your mouth, right the way down to the rectum and the anus. It's all covered in epithelial tissue. And as we've said, along the way, certain types of it are specialized. The goblet cells that we've had a look at, for example. So another important thing about this type of tissue, it's responsible for forming the majority of glandular tissue found in the human body. So glands are somewhere where hormones are released from. So it's very specialized in that it releases hormones. So we're going to have a look at that later when we do another unit on homeostasis. But most of these glands that release things like, well, you know what hormones are. You've heard about them in terms of the reproductive system, like estrogen, progesterone, testosterone, adrenaline, things like from the thyroid gland. So most of this glandular tissue is made out of this type of epithelial tissue. So it covers most of the body's cavities, as we said, the gut, the outer surface, the skin, but that's a different type of specialist compared to some of the ones that we've been looking at. And what are referred to as dead end tracts. So rather than the digestive system, which has got a hole at one end and a hole at the other, and things pass right through, things like the respiratory surface, which is, there's only one way in and one way out, genital tracts as well. So there's also things called ducts that open into these tracts. Duct just means a tube, really. So we've got certain organs like the pancreas that release things from ducts. They release hormones as well, down these tubes, so that sort of thing. So single epithelium might have several functions. So it might be structural, you know, forming a layer as a barrier for protection. There might be lots of different roles that it's carrying out. But the most important of these are the fact that it is interaction with the environment. So exchange surfaces, I keep talking about. So we're exchanging things like oxygen and carbon dioxide in the lung, all of the nutrients that we're taking on in the digestive system. So it's really important. If it's an exchange surface, you've got to have a mechanism for exchanging, getting the oxygen in, getting the carbon dioxide out. So things like diffusion, selective transport. So in the gut, we want to be taking in nutrients. We don't want to be taking in pathogens, absorption and secretion, things like that. So they will have very specialist roles. Okay. So underneath the epithelial layer, we've got this connective tissue layer. We talked about this bit last session, so I'm not going to dwell on this particular bit too much. So this is the connecting or supporting framework for most of the organs. The connective tissue is, remember we said, sparsely populated by cells. So it hasn't got that many cells compared to its extracellular matrix of proteins and fibers. And so most of it is fibers and proteins and not many cells. And what it does, it provides structural and mechanical support for other tissues, like the epithelial tissue we were just talking about, And it could be really important for the exchange of nutrients and waste in the example of blood. And it could be really important to help with regeneration and repair if tissues are damaged. And we said the main components of the extracellular matrix of the connective tissue was the fact that it had this matrix, loads of fibers, loads of proteins, and it had support cells. So we'll have a quick look at the ones that we looked at last session. And we said here, the blood, the matrix is the liquid, the plasma, and the fibers, actually, you can't even see the fibers in blood. Most of the time, they're dissolved because they're soluble. They only come out of solution during clotting. So if you get a wound that needs to be healed, the fibers come out of solution and start to knit together and block that wound. And then we've got the cells, the blood cells, and the function there is transport of oxygen, carbon dioxide to get released, and defense, against infection. And then the other one we briefly touched on was bone, which has got a hard and calcified matrix. The cells are just bone cells, which are osteoblasts and osteocytes and things like that. It's also got fibers made out of collagen in its matrix. Let's just check what that was in the chat. What would the fibers in the blood be called, please? They're called fibrin and fibrinogen, and we will have a look at them later on when we have a look at clotting. So you don't need to worry about them now, but they are important in a later unit. You don't need to, I really don't think you need that much detail now, but they're in solution, and they will come out of solution as and when they're needed based on signals from, well, inflammatory signals and wounds signaling, basically saying mend this wound. But don't worry too much about them now, but we will talk about them later. Okay, so the matrix then beneath the epithelial layer, the connective tissue that provides support for other tissues. It facilitates the exchange of nutrients, that's the blood, and waste and circulation, and also stores surplus energy as fat, because one of the tissue types is adipose connective tissue, which is the fat, really important for insulation. So it's made of specialized cells. We had a look at the macrophage, didn't we, last session, which is a specialized blood cell. We've got these fibers and potentially a gel-like substance, or it could be a liquid substance. So there's 6 major types, which are, this is all in the course material if you want to read more. There's loose connective tissue, there's cartilage, there's fibrous connective tissue, there's adipose, there's blood, and there's bone. And we specifically had to look at blood and bone. And the fibers are 3 types. So we've got collagen fibers, which is high degree of tensile strength, and it will withstand stretching forces, apply a lot of flexibility, quite important in terms of your skin, your bones, your cartilage, things like that. We've got elastic, which stretch under tension and return to their original length. They're thin and branched. And we've got reticular, contain collagen fibrils again, and join connective tissue to adjacent tissues, because obviously all these tissues have got to join together and often to form organs as well, which we'll have a look at later. So here are the examples. It's just a list really, but there's more information on the course materials of these different types of connective tissue. And if you have a look at this as the end of a bone, you get an idea of the extracellular matrix sort of structure that you can see here, holding everything together. Okay, so we had a little look at neurons earlier, and we are going to talk about them in terms of tissue now. So as we are going to learn later on when we do the homeostasis unit, there are 2 different systems in which humans can respond to their environment. There's the nervous system and there's the endocrine system, which is like hormonal. The nervous system is for really fast responses compared to the endocrine system, which is a bit more slow. So endocrine system tends to be longer, more permanent. So think puberty, pregnancy. I mean, obviously, like pregnancy is not forever. It might feel like it, but it's not. It's just a longer process. Nerve signals are fast, eyes blinking, pupils dilating because of light changes, things like that. So nervous system controls breathing, memory, intelligence, thought, and it involves nerves, which we had a look at a minute ago, made out of specialist cells called neurons. So there's a lot of information on this slide, and it's a little bit like, whoa, to start with, but I'm going to talk you through it. It's not as bad as it looks. It's just because it's so much writing, it seems bad. So here's your nervous system. That's all of your nerves, everything that you control via your nerves. It might be your muscles are controlled via your nerves. Some of your glands are controlled via your nerves. It could be thinking, doing maths equations. That's part of your nervous system. So we've got two parts to your nervous system. You've got your central nervous system, which I said earlier, down the center, clues in the name. So that's your brain and spinal cord. And then you've got your peripheral nervous system. Again, periphery, on the outside. So the brain is a huge subject on its own, and we don't go into massive detail about the brain on this course. It receives and processes sensory information. It can store memories, generate thoughts and emotions. That's a lot. Really, we're just interested in the receiving and processing sensory information, really, for now. What does the spinal cord do? It conducts signals from the brain, but it also controls reflex activities. So what do we mean by that? Well, let's just say you're in the kitchen cooking, you burn your hand. You don't want that signal to go to your brain going, oh, better move my hand, because by the time that happens, you could be really hurt. So the spinal cord will actually control a reflex activity. You hit something hot, you move your hand, you jump without thinking. So that's got an important control center role as well, but it's just a involuntary reflex control. So that's your central nervous system. Think of it as like the control center. Your peripheral nervous system is more like communication and control. So we've got motor neurons, think motor movement. That's how your central nervous system, CNS, sends messages to the muscles and your glands. Think muscles movement, yeah, motor movement, muscles. Sometimes it's sending a signal to a gland. So one type of, let's just say you smell some food cooking, smells really nice. You're not going to signal to a muscle to do anything necessarily, unless you're going to start eating, but you'll get a signal sent to your salivary glands. You start salivating, getting your mouth ready and your digestive system ready to digest and swallow food. So that's one time your motor neurons might send a message to a gland. Sensory neurons, these are things that take the message from your eyes, your ears, your taste buds to your spinal cord or your brain to process what to do with that information. So the sensory neurons we sometimes called afferent. I prefer to call them sensory because it makes sense because they come from the sense organs, but some people will call them afferent nerves. And your motor nerves, motor movement makes more sense to call them motor nerves to me, but some people call them your efferent nerves. Now, your motor neurons can be divided into two again. So remember, your motor neurons are the ones that say move. Muscles, usually they go to. Some of your movements are voluntary and some are involuntary. And the involuntary movements are divided again, fight or flight, and you've got rest and digest as well. So a reflex arc is a really good example. I know this is a lot of information now. We're going to look at it really in depth much later on. But as I said earlier, this is a really good example of something coming into your sensory neurons. It goes to your central nervous system, your spinal cord, which processes it, and then your motor neurons act on it. So Your stimulus, change in the environment, is picked up by a receptor, so a sense organ. It's not your eyes or your nose this time, it's your skin. And you feel hot, pain, ow. That signal then goes up the sensory nerve and into your spinal cord. There's something called the relay neuron there that relays the message in your spinal cord. It doesn't go to your brain. You don't think about it. It just happens really quickly. And then the message goes down the motor neuron to your muscle. Move your hand away so that you don't get burned. So that's a reflex arc. Don't worry too much about the details. We're going to look at that later on. But that's just to show you that all of these systems are working together. The peripheral nervous system, you've got the sensors, you've got the sensory neurons, the motor neurons, and the central nervous system all working together. So again, the peripheral nervous system, the tissue is nerves, they're bundled together into nerve cells. And obviously we've got these glial cells there as well. These are just structural support cells. They're in the central nervous system and the peripheral nervous system, and they maintain homeostasis, which we're going to learn about later, but that means keeping things in balance. So we keep our temperature in balance, for example. We keep our blood glucose in balance. So they're important for that role. So central nervous system, brain, spinal cord, the relay neurons we find in the spinal cord. It's how we get these reflex responses so we don't get burned, for example, connecting sensory neurons like your heat response, heat detecting signals in your hand, and to move your muscle away. So we're going to have a little bit of a closer look at these neurons. Let me just check time. Right, good, we're doing okay. So as we said, we're controlling how we respond to our environment through the nervous system here. And we've got these sensory neurons which relay messages from receptors to the spinal cord and the brain. So they're slightly different in structure to the generalized neurons that we had a look at earlier. So this is the one that would have picked up the hot signal in our fingers. Look, there's receptors in the skin. You can see it's got receptors on its end. And the signal will go down from the receptor to the axon of the nerve. And then we've got these endings at the other end, these synaptic endings, which will connect up to the relay neuron. And the relay neuron will take the message from the sensory neuron. This is inside the spinal cord. And the reason this reflex response goes through the spinal cord, as I said, we're not wasting time sending it to the brain. We don't want to think about whether we remove our hand from the burning object. We just want to do it as quick as we possibly can. So this has got a slightly different structure. Look, there's no massive long axon like we saw with the sensory neuron, because that's potentially got to go from our fingers to our spinal cord. So it's got to go the length of our arm, be quite long. This one is just inside the spinal cord. So we haven't got that big long axon this time. It's quite short. It's just inside the spinal cord. It's a relay neuron. It relays the message between the sensory system and the motor or movement system. Now, again, that motor neuron has got to go all the way down the arm to tell your arm to contract your muscle away. So it's going to be quite long. But at this end, we can see rather than the receptors that we saw here, which were in the skin, at the end of the motor neuron, it goes to an effector. Now, an effector is something that can affect or bring about change. And what's going to affect change in this situation is the muscle. I know it's muscle because it's stripy. Look, that tells me it's muscle. So we learned earlier, didn't we, that muscle's going to look stripy. So we've got our striated or stripy skeletal muscle there, which can contract to effect change or bring about change. That's what we call the effector in this situation. So there's our generalized neuron in the middle there. Here's our motor neuron going to the muscle that we just looked at, our sensory neuron with the sensors at the end, and our relay neuron, which some people call an interneuron, because we like to have 5 names when one would suffice, don't we? So interneuron just means like in the middle. Relay neuron I like because it relays the message. So the relay messages are found either in the brain or the spinal cord, but if it's a reflex response, really quick, it's in the spinal cord. They have a short axon because they don't need to travel very far. And then we see these big long axons in these other two nerve types, that neuron types that make up the nervous tissue. So we talked about the dendrites, didn't we, already? So we've got these dendritic structures at the ends here, looking very branch-like to form these branch connections. You can see as well in all three of those nerve types. And we've got these junctions at the end of the dendrites, obviously, because all of three of those nerve types are connecting with each other, these synaptic junctions. And as I said, some of these neurons can be really, really long, up to 2 meters, or, well, I don't know, 2 meters is quite a stretch, maybe not quite that long, but very, very long. And as we said earlier, when we were looking at the generalized structure, they've all got this myelin sheet, which is an insulation layer, helps speed up the processing, not really processing, helps speed up the sending of that impulse, because the signal can jump between these insulated sections to this node of Ranvier section. They've all got that structure, which will speed up that signal. And actually, one thing I didn't mention earlier is that myelin sheath. It's made out of something called Schwann cells. And it's really important because it stops nearby neurons interfering with each other. They can be bundled together, a bit like you can put electrical wires together in a big cable, and the signals won't interfere because they're not, you're not going to get sparks because each one of the wires is contained within its insulating tube. It's the same. You can get lots of neurons together in a nerve, and because they're insulated, they're not going to interfere with each other's signals. So the myelin sheath, this fatty layer, is made out of special cells called Schwann cells, and they wrap themselves around this axon several times. They're made out of a lipid, which is a fat or an oil, and it's called sphingomyelin, and it's a really good insulator against the flow of electrons, ions, sorry, not electrons. I'm thinking physics there, electrons, flow of ions along that neuron. Okay, so let's have a quick look. What's the main function of the nervous tissue in the human body? Which part of the neuron carries impulses away from the cell body or the axon? Have a look if you can answer those for me, and then we'll pop the answers up on the screen.
I didn't catch that. Could you try again?
Oh, so,
by the way,
sorry about that. My phone again. Every time I'm checking the time, see how I'm doing, it's activating Siri. Okay. So, function of the nervous tissue is transmit electrical impulses. The part of the neuron that carries impulses away from the main body of the cell is the dendrite. The axon's the main body there. Can a cell contain a cell in it? A neuron having a Schwann cell? Well, the neuron's actually got a Schwann cell wrapped around it. But that's quite a complex question, because we think that mitochondria were actually originally cells that became engulfed by a unicellular organism. And that's how our cells went from being simple bacterial cells to having structures within them. So can a cell have a cell within it? Yes, probably. And that's a very important evolutionary question. A neuron is kind of wrapped around the outside, I'd say. Okay, I'm going to skip those because I just want to move on to this because I'm running out of time a little bit. Outline how tissues form organs and give examples. So what I want to show you is the heart. So this is the heart. You can see top left hand of the screen there. And if we zoom in on the wall of the heart here, you can see we have got several layers. We've got an epicardium, a myocardium. We've actually got an endocardium as well. So what these different layers are made out of, we've got the epicardium, which is made out of epithelial and connective tissue, which is this here. There's pericardium there as well. We've got the myocardium, which is the muscle. And we've got the endocardium, which is the epithelial and connective tissue here. So remember we talked about endothelial tissue mostly forming like the outside or the surface of structures. So we've got endocardium here forming epithelial like tissues. We've got the muscle tissues, and then we've got several other layers. So loads of different tissue types. We've got, if we go on to the next slide, I know this is a drawing, but we can see, I like this drawing because it shows all the different structures. We've got all these veins, we've got the muscle, we've got the arteries, there's glands up the top there, we've got nerves, the vagus nerve in there, and it's a good illustration of how many different structures are making up this organ. So we've got supporting structures, we've got nervous and vascular structures. That's the nerves that are coming in yellow there. We've got the vessels, that's what the vascular means, of the vein and the artery. And then we've got all of these muscle of the actual heart, cardiac muscle, which has got these epithelial and connective tissue layers as well, making it up. So we've got the epicardium, the outer layer. It's made up of epithelial tissue and connective tissue. It's a thin outer layer of the heart. It's also called the pericardium, and it's called the pericardium on the diagram here. What it does, it protects the heart. It reduces friction as the heart beats. It's got blood vessels and lymphatics and nerves embedded in it. We've got the myocardium, which is the middle layer. This is the main muscle layer, specialized type of muscle. It's cardiac muscle. It's the thickest layer and the main muscular part of the heart, because heart's a muscular pump. It's really important because it contracts to squeeze and push that blood around the body. And it's got specialized structure for synchronized contraction, because obviously we want our heartbeat to be synchronized and regulated. And then we've got the endocardium, it's the inner layer. So this is the bit that the blood flows through, the blood that you're pumping out. It's specialized squamous epithelium, a type of connective tissue, as we know. It lines the chambers in the heart, covers the valves, which are important for stopping the blood flowing backwards in the heart. Why is this square tissue, like tile-like structure, important? It's a lovely smooth lining for the heart, so it proves smooth blood flow. We don't want turbulent blood flow, like we don't want it washing around in there like a washing machine. We want it to go in and out exactly where it needs to. It needs to flow really nicely through. It helps the blood to keep moving, and it stops it from clotting in the heart. So the supporting tissues, in addition to the ones that we've just looked at, we've got connective tissue forming the valves. We've got the nervous tissue, which will help to regulate heart rate. And we've got the blood vessels, because the heart itself is a muscle, so it needs to be supplied with oxygen. It can't just dip into the blood that's traveling through the heart to get oxygen. It has to have its own blood supply as well. And you can see that on the outside. We'll talk about that later on as well, because this is the one that gives you heart attacks if it gets blocked. It's really important. Okay, we are somewhere near getting towards the end of this section, this unit. We have looked at different tissue types in the body. We haven't quite had a look at stem cells yet, but we can talk about either epithelial or nervous tissue in reasonable detail. We've talked about how these tissues are grouped together to form the heart, and we've talked a little bit about regeneration as well. So you should be somewhere near able to start thinking about your assessment for this unit now. Have you been given your assessment yet by your tutor? I don't know where your assessment is. Your tutor's going to talk you through that, so you'll be getting it soon. But I'm just saying we've covered most of the material to be somewhere near ready. So as long as you've been doing the background reading and going through the course content as well, you should be almost in a good place now. to be able to handle that. I'm not sure. I'm not putting it up. It's your tutor who's putting it up for you, and your tutor that will talk you through it. But my point was really that we're somewhere near ready to make a start. Any more questions, guys? You have a few? Go ahead. I will do my best. Might not be able to answer all of them. I'm not ChatGTP. Whoa, that's loads. Right, let me see what I can do. Let's hope I can it's fine, don't apologize. It just came up in a big sort of, hang on, I'm gonna have to scroll down. It's not letting me look at it, I think, 'cause it's a big, I might have to drag down. No, it's not gonna let me. What are the fibers in the blood? Well, the ones that we're concerned about is the fibrinogen and the fibrin. What is the difference between macrophages and fibroblasts? That's quite a long answer. What I'll do is I'll pop that up for next session. I'll remember and do that. What are antigens? Right, antigens are structures on the surface of cells, or cells generally, or viruses maybe, stuff that we recognize as not being part of our body. So it's just stuff that our body goes, hang on a minute, that is not self tissue, that is not part of me. They can be proteins, they can be sugar molecules, but they're just stuff that our body goes, whoa, hang on, that's not part of my body. And then they are recognized by antibodies, which stick to them and flag them up for destruction, usually. ROS production. So ROS production is things, there's certain substances, ions, superoxide dismutase, things like that can cause lots of damage. They can cause cancer. They're not good. So it's stuff that we take, when we eat antioxidants, they mop up those active oxygen species. They can cause a lot of damage in cells. What is the difference between columnar and pseudostratified? So columnar is a column shape, and pseudostratified means like fake stratified, so fake layered. So it looks, it is kind of columnar, but it's not very regular. It's because it's got this sort of curvy structure. It looks, you've got a sort of top heavy cell, then you've got a bottom heavy cell. It looks like it's layered. So it's not quite a column, a nice, neat column. Difference between the dendrites and the synaptic endings. Dendrites is the branching, the synaptic ending is the very end. We will have a look at these in detail. They're very specific structures. We'll look at them in the homeostasis unit. Oh yeah, I'm on my touch screen. That's good. Let's see if I can move it that way. No, it's not going to let me. I think it was something about the difference between muscle. Why is voluntary muscle stripy? It is just the layout of these actin and myosin fibers. It's really contractile, so it's got to pull in a lot. It's the very big, powerful muscles. So it's just the layout of this actin and myosin, which is like this. Imagine if it looks like this, and then if you overlap it, it's more dense area where my fingers are overlapped. So it looks like a stripe under the microscope. That's all that is. I think that's most of it. What was the one that I was going to look up and give you a better answer for than I can do off the top of my head? Cell differences, macrophages and fibroblasts. Yeah, I'll look that up and give you a more detailed answer than that. I can't answer that very clearly. They're very similar. Okay, so any more questions? Okay, guys. Fab. Well, I will see you on Tuesday then. Bye, everyone.